Yeah,
Hi, how are you?
all good, sir.
Good.
Oh,
There we go.
yes.
All right. Well, welcome. So, what's going on new?
New? I don't know. I've been looking up papers about tissue modeled with tensegrities, and there aren't very many as far as I can see. I'm supposed to do a review. How do you do a review with three papers plus your own? I'm just not sure.
Well, you could go in depth in each one. I mean, that's
That's true.
It's not
Mhm.
not a review at that point. It's more, well, it is a review, but it's more focused, I guess. Are you sure there were only 4 papers, or are there like things
Yeah,
that aren't called tensegrity that are kind of similar?
I have one where they, well, it was working really great until the tensegrity did its thing, which is a soft mode, and it just all of a sudden changed.
Yeah.
So, not too sure about that paper because the model worked fine until it didn't.
Yeah.
I don't even know what you would say about that. Like,
I don't know.
yeah,
I mean, you could well, you'd have to evaluate the results, probably make sure that they didn't publish something that was just kind of like a failure mode, and they said, "Oh, that's interesting."
yeah. So then you'd have to redo the model. Pardon the dog. I have a dog.
Oh.
She likes her squeaky toy, and it's annoying. But anyway, so she's a puppy.
Yeah.
Yeah. And now here we're done. Anyway, good enough. That's what I've been working on.
All right. Well, that's good. Thanks for the update. And then Lilith, how are you?
Good, sir. Sir, I've been looking at some of the GitHub repositories of the DevoWorm and OpenWorm and searching about what are the previous contributions and all. So I got to find out some of the, this structural connectomes of, the C. elegans. So I've tried to load it and see how's, there are like some few good visualizations tool also. So I've been looking at them, and I'm interested. I've seen some good paper recently. I don't know. I have to go through it a little bit. It's a little complicated. So what they have done is they have done this functional connectivity, and it got published in Nature 2023, I guess. So it's from Professor Lehner's lab from like Princeton. So they have seen that. So I'm just reading this, that the neural signal propagation, how it's happening for C. elegans and all. So it was kind of little interesting because so although we have like wired structural connectivity and the connectomes are like very very much wired entirely, but there are some activations and there is some signal propagation, although there was no like wiring and all. So that was a good line of work. I wanted to, go a little deeper and understand. I'm trying to see the code of them. So it's open source, and they said all the figures and everything can be reproduced. So I'm looking at their work and a little more deeper into like what we can do.
Okay. That's good. I don't know if I guess Mahul said he might be here, but I don't think he is going to come. He wants to do a presentation on some of the hypergraph work he was doing last year. So if he comes next week, we'll be able to see that. But yeah, that sounds like a good paper. Can you send that paper along to me at some point? Like
Yeah, I can send it just, I can just attached, here, or I can attach in the Slack, it could be great.
Yeah, actually, if you could put a link in the chat here, it would be good. Or something like that. Right. So Aniketh, hello. Aniketh is there. If you want to introduce yourself. There's Morgan. Hello, Morgan. Dick.
Good morning.
Oh, good morning. So last time, Lilith was talking about this comparison between artificial neural networks and biological neural networks, and I brought up the paper that we mentioned in the meeting, where that we had done in 2021 in our group, where we looked at a couple of different criterion differences between the two types of model, or at least real brains and the model of a brain. And so, we have to think about kind of those differences, because people want to sort of, they want to recapitulate biological brains. They don't want to, they want to make artificial models better, but ultimately the goal is to sort of emulate a real brain, or emulate what things a real brain does that are interesting. So, this is nice to have this sort of comparison to lay out the different things that people might focus their work on to, move towards that goal. So this paper here, I think this is in neural computation. Yes, this is a neural computation paper. The title, and I'm not crazy about the title, but whatever. Modern Artificial Neural Networks, Is Evolution Clever? And this is, I think it's by a German group, or also, there's an American in there. But anyways, this is about looking, and they don't talk about evolution much in the paper. They just kind of talk about it at the end. But the idea is that evolution produced biological neural networks. Therefore, it's maybe cleverer than we can, or more clever than we can have in terms of artificial neural networks, and something that we kind of construct from this metaphor that we're using. So I'll go through the abstract. There's a lot of detail in the paper, so if you're interested in looking that up, it's actually this neural computation paper from 2023. So the abstract reads: machine learning tools, particularly artificial neural networks, or ANNs, have become ubiquitous in many scientific disciplines, and machine learning-based techniques flourish not only because of the expanding computational power and increasing availability of labeled data sets. So this is where we have these, we have this ability to sort of make these models work through a lot of training and a lot of computational power. Now, of course, biological brains, one of the sort of the benefits of that, or emulating the way it does computation, is it doesn't take that much energy at all, at least comparatively. And you don't necessarily need a lot of information or training to do things. There are a lot of things that biological brains can do that require very little interaction with the world, at least relative to the sorts of training regimes we use in ANNs. So that's, this is why they're flourishing, but also because of the increasingly powerful training algorithms and refined topologies of artificial neural networks. So this is where, we have these topologies that we can, we can select for certain weight schemes, and we can get good topologies that are optimized for learning at least certain tasks. So one of the drawbacks, of course, to ANNs is that we have a hard time generalizing them to other tasks. So we can really, use a lot of data, train them for a single task, but then when it comes to generalizing to new things that the network hasn't seen before, it becomes harder. And so, but we do have powerful training algorithms. So this is one of the, the great breakthroughs of the last maybe 10 years or 15 years. Some refined topologies were initially motivated by neuronal network architectures found in the brain. So sometimes if we use neuronal network as a term for BNNs, we, have convolutional artificial neural networks, which are like neuronal network architectures in the brain. Later topologies of neuronal networks departed from the biological substrate and began to be developed independently, as the biological processing units are not well understood or are not transferable to in silico architectures. So again, we have this sort of abstraction problem where we have maybe the single unit that does a single computation. And then we could have, maybe a single unit with multiple compartments. But we don't really use, say, like all of the things that we have in a neuron. That is to say, we don't have like a molecular scale set of processes that have an output. We don't have, biophysics. We don't have action potentials that produce an output. We sum inputs and produce an output, but those are all computational in nature. And so, you could argue, well, the brain kind of does this anyways. The lower level details don't matter. But obviously, there is something there that's going on that we don't know if it really matters or not. We just say that this is basically what it's doing. That's kind of the point of abstraction when you take a real world system and you say, this is basically the way the system works. If we were to take a pendulum and we were going to build a physical model of the pendulum, we were going to build a computational model that we would put into some coding language like Python, we would take the sort of the, most basic elements of that relationship of the pendulum and put them into an equation and model that equation. But, there are all sorts of stochastic fluctuations that might account for variations in the pendulum that we don't add into that model. And so you see a difference. And so you might say, well, why don't we just add in, some sort of stochastic signal into neural networks? And I think, even if we did that, we'd be missing other processes that are kind of contributing maybe additively or multiplicatively to the output that we see in biological neural networks. Anyways, that's an aside. That's something to think about in terms of, what the differences are. In the paper we did from 2021, we didn't even, I don't think we talked too much about that. I'd like to revisit that though. In any case, and so, we started with these kind of models that were abstracted away from biology, but then we were sort of, we kind of thought, well, this is basically what's going on in the brain. And of course, this was back in the 1940s and 1950s, where you have the McCullough-Pitts neuron, and then later you have these early connectionist models. And people, basically use the general principles of the brain, at least as people saw them then, and said, well, okay, we can build a model from that. And so, we have these models that are sort of semi-biologically inspired. But of course, later topologies of neuronal networks departed from the biological substrate and began to be developed independently, as the biological processing units are not well understood or not transferable to in silico architectures. So what that sentence, I guess, means is that, we have this sort of model and we kind of think, okay, this is basically what's going on in the brain, but now we need to make it, we need to add some technical details. And so as we added in technical details, they departed from whatever was sort of similar from the biological substrate. And we can't seem to transfer some of the key sort of details from biological neural networks to artificial neural networks. In the field of neuroscience, the advent of multi-channel recordings has enabled recording the activity of many neurons simultaneously and characterizing complex network activity in biological neural networks. The unique opportunity to compare large neural network topologies, processing, and learning strategies with those that have been developed in state-of-the-art ANNs has become a reality. Okay. Then, so we have these tests, these statistical tests we can use, of course, that will compare something like, some structure in the visual cortex with maybe like an artificial neural network that does feature selection in a database of images. So there's a biological neural network doing something similar to what an artificial neural network is doing, maybe on a benchmarking task. And then there's statistical methods where you can compare the networks. You can extract a neural network from the visual cortex, like somewhere like V1 or V2, and then compare that with a neural network that's processing images and doing feature selection. So they're doing the same task, but they're, of course, they're different networks. They're doing things differently, and we can actually see how well they're correlated and how well they are related to one another. But that's kind of limiting because it doesn't tell us a whole lot other than that they seem to have similar behaviors inside the network. Everything else is really kind of a black box. So that's one of the challenges, is finding these kind of comparative techniques. So the aim of this review is to introduce certain basic concepts of the modern artificial neural network, corresponding training algorithms, and biological counterparts. The selection of these modern artificial neural networks is prone to be biased. They exclude spiking neural networks, which are these neural networks that have like a biophysical component. So they those are different in terms of how what the outputs look like and the sort of activation functions they use and so forth. So this is kind of an overview of these artificial neural networks as they see them. They talk about the early days of artificial neural networks with McCulloch and Pitts, and then Konrad Zuse, who developed the first computers, Marvin Minsky, then the perceptron with Rosenblatt and Whiteman, and they were able to kind of move from that to this sort of connectionist view. So this is all kind of going through this history and talking about how our artificial models first arose and then how they diverged from the biological reality. And some of this was, or a lot of this was because of the technical challenges. So, you create a simple model, you try to improve that model, improve its performance, and then you use, of course, computational techniques, and you diverge from the biology. Granted, at the time, computational biology and theoretical biology were in their infancy. So we didn't have some of the tools maybe we have today. Although I would say that there hasn't been a huge amount of work in this direction. That's really, obviously, because we don't really have the tools to do really good comparisons. So that's that paper. I don't know if Lilith had anything to say about that, or if you wanted to comment, if anyone else wanted to comment on that.
So that was an interesting line of work, sir, because it focuses on the topology and how the structures are actually they are meant and all. And although they are excluding the spiking part, it will help to understand how it has been emerged from the smaller set of neural networks to that of what are we currently facing challenges. So it could be a nice read if I go detail into it.
Yeah. Okay. Anyone else? Okay. Well, welcome, Soham. I see Soham is here.
Hey, hi. Sorry, I got a bit late.
Okay. Did you have anything to say or update us with?
No, I was just going through a few papers. I have a few ideas. I'll concretize it and let you know in the next meets.
Okay, sounds good. Okay, so then the other paper I want to talk about today is this paper by Manicka and Levin. So this is a Michael Levin paper. This is a preprint that he's put out on field-mediated bioelectric basis of morphogenetic prepatterning. So this is on some pretty hefty sort of bioelectric developmental stuff. So this is really kind of getting deep into this sort of topic. So, if there some technical details that we don't understand, this is something that I don't know if I can help with, but we'll see. And so, of course, Michael Levin has done a lot of stuff with this bioelectric communication. So, neurons, of course, have bioelectric communication. We're familiar with, with action potentials, and we're familiar with gap junctions. And we know that, neurons express different ion channels. Different neurons express different mixes of ion channels. And we have, tools to look at that and say something about the bioelectric potential of a neuron. Well, of course, neurons come from stem cells, and those stem cells, you get different differentiation signals, and you can get things like muscle or other types of cells. And so muscles also have some bioelectric potential, and so we know how to measure that. And then, of course, we have other types of cells that are somatic cells, and those cells have, as it turns out, bioelectric potential. They express ion channels, just like neurons and muscle. Even stem cells express ion channels. And so, so they have this sort of potential. And just because neurons have an action potential doesn't mean that other cell types don't have some sort of trace that you can measure. Now, it's not going to be a spike like you see in an action potential, but it's going to be something else. It's going to be a different type of signal. Nevertheless, we can use that signal to understand what cells are doing. And so Michael Levin has picked up on this tradition of, there's a tradition that goes back maybe to the early 20th century of people looking at bioelectric signals in cells and thinking that that plays a really important role in development. So in development, we've talked about gene expression, we've talked about physical forces, and bioelectricity, of course, plays a role, but that's actually less explored in the modern literature anyways. Every time you see a big nature paper, they don't really talk about bioelectricity as a topic. So keep that in mind as we go through this abstract. So the abstract reads, bioelectric communication among cells plays an important role in numerous morphogenetic processes. Computational modeling and experimental modulation of these processes have largely focused on a discrete, localized view of cells in intracellular networks. So, we're kind of thinking in terms of computational models, and these computational models, like our artificial neural networks, are discrete units that are linked by edges that have some weight, and that's the way we usually model a lot of networks and models of cells and intracellular networks of different types. So a lot of these, we have to use these kind of models to understand spatiotemporal patterns in biology. We saw a paper last week where they did things with next-gen sequencing. So they were looking at what single cells are expressing certain genes and in different gene families. And then, they were able to produce a map where you knew what cell was what and what position it was in, and you could get a sense and see elegance, at least, of spatial patterning. Well, a lot of times we don't have the luxury of understanding spatial patterning in that way. So there is like the model that you see in Drosophila, where you can see the striping in a syncytium, and you can measure that with some sort of microscopy. In this case, what they're interested in are these networks or the spatial variability with respect to bioelectricity. And so you're getting these spatiotemporal patterns of resting membrane potential, or what they call VMEM, within tissues. So you're getting this membrane potential of the cells. You can measure that. You can use different types of photovoltaic dyes. You can use different types of electrodes and get that measurement, and then see what that looks like across the tissue, and maybe what role that plays in collective organization of the cells. Okay, so here we characterize novel contributions of the electrostatic force field, a continuous and pervasive entity existing within living tissues and modulating intracellular interactions. So they're talking about this electrostatic force field, which sounds a little bit like they came up with it in Star Wars, but it's not. It's this force, where all the cells sort of have this electrostatic potential, and it's this emergent phenomenon. And so, there's this idea, of course, that, in there's this idea of, or this notion of morphogens, which are these organizers, and they play a role in morphogenesis. In this case, what they're kind of arguing is this electrostatic force field plays a role in morphogenesis, and it's very similar to the role morphogens play in chemical morphogenesis. So, so this electrostatic force field is a continuous and pervasive entity existing within living tissues and modulating intracellular interactions. So this is a continuous model. So if we were to model this electrostatic force field computationally, we'd have to choose a continuous modeling framework to do this, and we'd also have to account for sort of its pervasiveness. And so this is, of course, a very different model than what we, from a network model. So that's kind of where they're going with this. And Mike Levin has published papers on this in the past, so this is nothing new. He's done a lot of stuff in model organisms that have demonstrated a lot of this. Okay. We consider a minimal but biologically plausible non-neural bioelectric network model endowed with an electrostatic field where the VMEM, or that potential, membrane potential, of a cell is regulated by the average strength of the surrounding field via negative feedback. So that resting membrane potential is, of course, sort of the baseline, and then that's regulated by the strength of the surrounding field. So it's almost like you have a membrane potential in one cell that's influenced by the membrane potential of other cells. And that's how you have this, it's almost like if you were to have a cellular automata model and you had a neighborhood of cells. So the cell in the middle is influenced by its neighbors, and then every cell has neighbors surrounding it, and those neighborhoods exist in parallel, and there's this sort of feedback, or negative feedback in this case, between the focal cell and its neighbors. And then this happens in parallel across the field or across the tissue or whatever entity we're talking about. A detailed examination of this model revealed that the field systematically regulates key statistical and dynamical characteristics of spatiotemporal configurations of VMEM patterns such as complexity, dimensionality, and causality, leveraging a mechanism akin to synergetics, where it simultaneously forms a lower dimensional projection of the pattern and enhances the causal strength and distance among its components. So synergetics, we'll get into this. I think they'll probably review this in the paper, but this is a form of complexity theory where they have formal models. We'll get into what that is. And then we have this lower dimensional projection of the pattern being enhanced by the causal strength and distance among its components. So I don't know exactly what that means, but basically you get this sort of, this projection of a pattern through these dynamics, and then there's this reinforcement that occurs across the tissue. Moreover, the almost instantly penetrating nature of the field endows the system with a unique amenability to self-organize complex positional information patterns following a transient stimulation of just the boundary of the tissue. So when we talk about this complex positional information, we're talking about cells kind of having a location, and obviously cells don't carry around a map, or they don't have, maybe they have some sort of accounting mechanism, but it would be kind of hard to implement if the cells are moving, they're migrating and things like that. So you have to have this sort of system of sort of co-registration with the rest of the tissue. So cells know where they are in the anthropomorphic sense. So yeah, and then he taught, okay, so that's that part. And so then they proposed that this this self-organization of complex positional information of patterns has this organizer-like property that suggests new avenues for modulation in biomedical contexts. So this could be applied to some sort of biomedical context or bioengineering context, synthetic biology context. We used automatic differentiation-based machine learning methods to optimize signals from a symmetry-breaking organizer region, which would induce development of a vertebrate face pre-pattern. So they're looking for this face pre-pattern. They want to sort of simulate this face pre-pattern. They want to use, so they're using differentiation-based machine learning methods. They're basically trying to take signals from this region of cells, this bioelectric pattern, or these signals, and then develop this pattern in silico, as I understand it. A detailed spatiotemporal dynamical analysis of a pair of such models with differing degrees of field sensitivity revealed very different collective coordination strategies with which the model developed the pattern, namely a preformed mosaic mechanism and an emergent stigmergic mechanism. So this is, kind of arguing that there is this sort of, maybe this preformation aspect that's initiated by bioelectricity. And then there's this emergent stigmergic mechanism, which stigmergy, of course, is where you have a system where there are interactions, and then there's a trace left of those interactions, and that trace then forms these patterns. So if you think about like ant colonies, when ants walk around a surface looking for food, they drop a pheromone trail. And those pheromone trails then are reinforced by other ants following along. And so you end up with these ant trails that form this emergent structure, and it allows ants to find food. They're able to navigate a space. They're able to basically lay down the precursors for positional information and guide other ants to the food source. And this is done more efficiently through these pheromones or the stigmergic set of interactions between ants, where one ant will lay down a pheromone trail, another ant will pick it up and reinforce that trail, and so forth. So this is what he means by sort of this emergent stigmergic mechanism. It's some sort of mechanism that reinforces an initial pattern. So it may be a preformed pattern. It may be something that kind of emerges from the most likely interactions and then is amplified accordingly. The stigmergic pattern, in particular, recapitulated several key features of the developmental sequence of the bioelectric facial pre-pattern observed in frog embryos. So they're able to see this analogy with frog embryos. These results highlight the promising potential of the electric field per se as a facilitator of collective patterning, providing a kind of master regulator interventional target for applications in regenerative medicine and bioengineering. So this, he's been working into this direction of trying to, find ways to build in these regulatory systems for things like synthetic biology or bioengineering, where you're trying to pattern different types of higher order tissues, or patterns on tissues, or organize things like faces from a group of tissues. So if you think about like in tissue engineering, when you want to achieve some sort of pattern, or some sort of if you wanted to build a face, or if you wanted to build an organ, this would this kind of mechanism would work. So I know that Morgan in particular is interested in organoids. You could think about the problems that we have with organoids as an example of how this could solve that problem, namely that organoids will form these structures. We have to do a lot of careful bioengineering to get those structures, and then we have to sort of combine different organoids together into these assembloids. And so, one of the things we could do, of course, or one of the things we don't do, is we don't think about the bioelectric aspect of it. I don't think anyone really, aside from maybe recording bioelectricity, they don't leverage it to sort of sculpt these organoids and use them as instructive signals for forming patterns. I'm not aware of any such research. Okay, so the biological field is this concept that they're using. This goes back many years, and there's this idea, more commonly known as the morphogenetic field. In modern developmental biology, it expresses informational regional relationships, the prototypical example of which is the morphogen gradient, or the French flag model, and is often referred to, or as it is often referred to. And then he brings up Turing, who, of course, was trying to find some sort of substrate invariant decision-making system, and he had this biochemical basis of such a system mimicking the self-organizing properties of embryos. So this is in reference to some of the work he did not only on chemical morphogenesis, but then kind of transferring that in the direction of artificial intelligence. And I'm not familiar with what these references are, but basically gets into that history of that work. Now, I know that Dick has mentioned in years past that he doesn't believe in morphogenetic fields, I guess, but this is kind of where, the model that Levin is using. So, I mean, it's not without its controversy. It's not without its sort of taking a position on that. So that's basically what we're doing here. So one important set of biological regulators concerns ionic signaling. All cells, not just neurons, communicate electrically in vivo. So this is where you have this literature on ionic signaling and how that can play a role in patterning. Bioelectric properties in tissue have been shown to underlie prepatterns regulating the formation of complex structures such as the vertebrate face, axial patterning of the anterior posterior, dorsal ventral, and left-right axes. So it's front to back, side to side, and top to bottom in an organism like C. elegans or in an organism like a lizard, where they have those are the orientations of the spatial axes, the location and identity of specific organs such as the eye, and the egg ovary system, patterning of epithelia such as skin and feathers, and size control. So last week we talked about feathers and scales and hair follicles. And so those, of course, form from maybe like a stem amnio common ancestral sort of set of processes that form this invagination in the epidermal layer, and then you get these different structures that can form later. So in this case, though, he's arguing that there's this epithelial mechanism that also plays a role in these types of things. And it's something that we don't usually talk about when we talk about these things. Okay. So bioelectricity is also important because there are a number of gene expression and epigenetic targets that lie downstream of these changes in biological or in bioelectric state. So basically, bioelectricity sets up, at least causally, some of these changes in gene expression and epigenesis. So, we think of those things as sort of maybe autonomous, or we focus on the environmental signals if we do something like a perturbation study, which we saw last week with the feathers and scales paper. But in this case, he's saying that the bioelectric state is actually one of the main causal factors in this. So this is their model here, and it does look kind of like maybe like a cellular automata. It's really just an array of cells where you have this continuous model. So you have this blastula on the left, which is this clump of cells. And this is a blow up of like a piece of the blastula. So these cells here are represented by cells in this array. And we have the individual cells. We have the electrostatic fields, which are these red lines. We have the ion channels, which are these black little squares on top of the cells or on the sides. We have these gap junctions, which are these gray lines. I don't know if you can see. But basically, we're representing this electrical sort of pass-through communication at gap junctions. We're representing ion channel communication across the membrane with these black squares. We have this electrostatic field, which is generated in this, by the balance of ions in the red lines, and then the cells themselves. So the cells interact based on these different sources of electricity and field generating phenomena, and then we get this field grid, which is these gray dots, and they just form this grid. So all of these cells are related by this field, and the electrostatic field, of course, affects the local cells, and this happens in parallel. So if you move from one end of the field that he specifies to the other, you get different electrostatic fields that interact. And so this is where we get this, this sort of emergent collective behavior is from all of this happening in parallel. And so this shows figure 2 static snapshots of the electrostatic force fields that are profiles of the model where when a single cell is active. So if we look up at the top left image, we see that single cell activation is happening in this top set of examples, and then tissue activation is at the bottom set of the examples. So let's go to the top left where we have short field range. So we have the cell here, it's in the middle, and it has this force field that only involves one cell. So that's a localized effect. Then the field range expands. So it expands from the just around the single cell to a neighborhood of 2 cells deep. So it's like 2 cells beyond the single cell in every direction. And so, this is kind of like a cellular automata neighborhood where you have neighbors and they have a state, and then you're, kind of integrating that information, and you're doing that locally in parallel. So this intermediate field range includes 2 cells out from the center cell, and all these cells are in this field. And then imagine that every cell has an intermediate field of the same size, and you can see where these interactions can occur. So again, this involves more cells, and then the long field range is this entire array of cells, so the field encompasses this entire array of cells. So it really depends on the field range. That depends what the interactions look like. You can sometimes have only one cell in that field range, multiple cells in the field range, and having this happen in parallel, and then a long field range where there's a long cast signal that casts over many, many cells. And these things can happen in parallel, but the reality of it is that the long field range is sort of this maybe global signal. So this is, when the cell is active, top row, and then when the entire tissue is voltage patterned, bottom row. So in the top 3 examples, we have single cell activation, which is where the single cell is active. And then at the bottom, the entire tissue is voltage patterned, meaning that we have these different sort of orientations in the field, and that leads to these spatial patterns. So for short field range, we see this here, where we have this pattern which is differentiated on the left side or on the right side. There are these differences in the field, the orientation of the field. And then intermediate field range, we kind of see the same thing. And then in the long field range, it kind of focuses on this area here. So I'm not really sure what the lesson is from this. But yeah, so the darker shaded units in this tissue activation model represents more hyperpolarized potentials. So these cells have different potentials, and then it leads to patterning. So you can see that the ones in black here are more hyperpolarized than the ones in gray. And of course, that's where you get this difference in polarization. It's not like a action potential, but it's like a kind of a similar principle where there's an activation versus a non-activation. And so that's kind of what we're looking at in this array. And then they have this 11 by 11 model that generates patterns. A short field range is pretty much, this block. Intermediate field range has some pattern formation on the edges. And then the long field range, you get this gradient of colors here, which I guess is where they're generating different patterns. So at the top, we have weak field sensitivity, short field range, intermediate field range, and long field range, all having different sorts of sensitivities and pattern formation potential. Then on the bottom, we have strong field sensitivity, which is where we have high W and low B, which are the top, we have low W and high B, and they define these parameter values in the paper. Just know that there's a difference between weak and strong field sensitivity. You get this, again, with strong field sensitivity, short field range, it gives you a little bit different result on this pattern, but most of it is the same. Most of it is the same. Intermediate field range, you're starting to get this broken up pattern formation. You're starting to get these centers. And then long field range, you just see something in the middle with some decay at the edges. So that's kind of, and then these, again, these darker shades represent more hyperpolarized potentials, meaning, of course, that these cells are more differentiated in the long field range and in the intermediate field range. So that's basically what they're trying to argue. So basically, the idea is that these electrical potentials are activated in cells. That activation then leads to gene expression, and then gene expression changes lead to these sort of pattern phenotypes. Differences in gene expression can lead to differences in morphology or in other types of signaling that are happening. So it's basically the sort of the beginning of the causal chain, I think, is what they're arguing. So in summary, these observations show that endowing a conventional bioelectric system with an electrostatic field enhances its patterning capacity, as evidenced by an increase in the complexity of VMEM patterns with an increase in the ability of the cells to sense changes in the field surrounding them. So it's actually that there's this complexity of VMEM patterns in a field. This allows for cells to sense changes in the field surrounding them because the field becomes more heterogeneous and you can detect differences. And so cells will do this sort of, it's almost like a psychophysics where they can detect changes across space at some resolution. So if you have like a sharp drop off or a gradual drop off, that's something a cell can sense and behave accordingly. So this complexity of the field is very important, and they get into much more detail in the paper on this. So they actually have this complexity measure. I'm not really familiar with it, but this is the Tononi-Edelman-Sporns complexity measure. So these are 3 people who are involved in a lot of different areas of research. I think Tononi is involved in consciousness research. Edelman is a big name in biology and in other in brain science, and then Sporns, of course, does a lot of stuff with brain science, but also complex networks. So they have this complexity measure TSE, which they'll be using. This is an information theoretic quantity. So Tononi's integrated information is kind of related to this, and it quantifies the trade-off between integration and segregation of the VMEM patterns at multiple spatial scales. So it's looking at sort of the total sum of mutual information between bipartitions of the system covering all partition sizes. So they kind of do talk about this in the methods, but basically it's a way to understand the complexity of these fields, the heterogeneity of these fields, and to get a single measure of this, get a single parameter that tells you something about the information of these fields. When the tissue is fully integrated, the cells tend to behave synchronously as a single whole, and when it is fully segregated, the cells tend to behave independently. So if we look at the tissue being fully integrated and the cells behaving synchronously, or when the tissue is fully segregated and the cells behave independently, TSE is 0. So when we have basically total homogeneity or total dissociation of the cells, TSE is 0. When we have this sort of heterogeneity of a tissue, then the TSE is non-zero. And that's what we're interested in. We're interested in maximizing that complexity measure in whatever way that it happens. So complexity is maximized when the balance between the opposing forces of integration and segregation are maximized, which is precisely what a good patterning mechanism must possess, since a pattern by definition requires a mixture of correlated and uncorrelated regions. So this goes back to our local versus global distinction. So locally, we expect things to be correlated, especially in a spatial array. And the reason for that is because local things should tend to behave together. They're signaling each other, they're communicating, they might be of the same tissue, they might be doing similar things in space, and so those things should be correlated. We should also have, over different length scales, decorrelation. So a cell, say in the center of the array, will look different from cells at the edge of the array, just simply by virtue of them being in different places, being in different populations, being far away from one another. And what they're arguing here, though, is that it's not a simple case of decay from a source. It's where you have these regions that are sort of correlated, and then regions that are uncorrelated. So you might have cells that are all in the same area that are uncorrelated because they're fundamentally different in terms of the signals that they're receiving or the signals that they're emitting from other local areas in the array. And so you'll have these correlated and uncorrelated regions. And this is, where you have these opposing forces of integration and segregation. Some parts of the tissue are being integrated, some parts are being segregated, and that's how you form a pattern. You have, if you have a mix of things at random, some of those things have to be organized into clumps. Some of them have to be broken up. So you have to suppress some cells so that you can have clumps. And that's how you get this sort of patterning. Then they talk about not local long distance signaling. So they talk about these non-local causal interactions among the cells constituting the VMEM pattern. So long distance signaling plays a crucial role in morphogenesis, facilitating spatiotemporal coordination among the parts of the developing system. Some of the major sources of long distance communication that have been so far identified include diffusing morphogens, and this is from, of course, chemical morphogenesis, where you have these signals that diffuse across your pattern forming surface, and that these morphogens interact and they the gradients collide, and you get these striping patterns or other types of boundaries. Cellular protrusions, tunneling nanotubes, macrophage networks, as well as bioelectric signaling. So all these things are kind of known as these long distance communication sources. So you get things that communicate locally, but also long distance. And so if you have long distance communication, you can have things that are intermediate to global and local pattern formation. Electric fields are also known to play a role during development in the form of cell migration, or what we call galvanotaxis, proliferation, and modulation of ion channels. And so the question is, could the electric field also facilitate direct cell-to-cell communication, especially at long distances, even if it is weak? So could we have this weak electric field facilitate direct cell-to-cell communication, but at long distances instead of locally, like what we are supposing with our neighborhoods and our local interactions? In the context of our model, could the field catalyze the interactions among the components of the VMEM pattern despite being orders of magnitude smaller, about 1% than VMEM? So in answering this question, they considered the measures that express the degree and length of causal interactions between cells facilitated by the field across space and time. So they define causal strength and causal distance, and that's talked about in the methods. Causal strength being the absolute value of the derivative of the DMEM, which is the VMEM, which is this potential of 3 representative target cells of the tissue at representative time points greater than 0 with respect to the VMEM of every cell at t equals 0. So basically looking at the derivative of this VMEM parameter for, you know, a subset of target cells looking forward in time and versus looking at every cell and its potential at time zero. So what is the time evolution of the signal, and what is the time evolution locally of the signal? So the causal strength should be, if something happens, you know, T0 is sort of your baseline, and then you move out in time and you say, locally, what is the causal strength of, you know, how is whatever's happening at T0 affect what's happening later, and what is the strength of that causal relationship? How strong is it that the VMEM at one point is affecting the VMEM at another point, and specific to certain cells? So that's causal strength. And then causal distance is the distance corresponding to the highest causal strength averaged over target cells in time and expressed in units of distance, or m, which I guess is meters, which, you know, you can use something a lot smaller than that, but I don't know. Yeah, anyways, it's a unit of distance. Causal strength quantifies the sensitivity of the VMEM of a cell to small changes in the VMEM of another cell, separated in space and time, and causal distance indicates the distance at which it occurs. So causal distance, of course, is looking at this spatial aspect. And so this is where we want to average over our target cells and then in time and look at the units of distance. So it's basically like a spatial correlation between the cells in the field. So if you look at the center cell, you can look at rings around that center cell and you average over that ring and you say, what is the causal distance? How is this focal cell in the middle of the array affecting cells at different distances outward? And so again, if it's at random, you should see this decay over space. From the center cell, the causal distance should decrease as you go outward. But you may not see that in a highly patterned shape or highly patterned field. You might see variation across the field. And so that's what he's getting at with causal, or they're getting at with causal distance. And then this is different than causal strength, but they're related. So this is the figure 6, causal strength and causal distance as a function of field range. These red lines here indicate the CS values, the causal strength values, and are plotted against the left axis. And the blue is the causal distance values that are plotted against the right y-axis. So you see they do one of these graphs where they have two different y-axes, causal strength being on the left, the red, causal distance being on the right, the blue. And you can see that in these different examples, they change the weights and they change the threshold. And for different weights, as you get to higher weights, you get these like these enhanced causal strength and causal distance measures. In this example here, you get it earlier in terms of the weights. So you get it for lower weights. And so, you can see the variation here. Basically, this is an example of weak field sensitivity, basically where causal strength and causal distance are flat. And then this is an example of strong field sensitivity here, where you get these enhanced values. Okay, so again, they go back to sort of this idea of an organizer, and that's what they're trying to get at. And they ask, could the electric field facilitate such patterning to transient stimulation of a small part of the tissue? And I guess the answer is maybe. Moreover, several known morphogenetic patterning processes in embryos adopt a linear and direct decoding strategy, where most of the information about the pattern is supplied by the morphogens, or deduced using relatively simple interpretation processes, such as the French flag, which is where you have these stripes in a field that get formed, and the clock wavefront mechanism, or the clock-in wavefront mechanism, which is another model that involves the timing of waves. And so they're comparing it to these more traditional models. And they ask, could there be a more nonlinear and complex strategy where the initial information supplied by an organizer-like entity is coded, and the system actively decodes it to generate the full pattern? So is this, where the organizer is kind of giving it a template, and then everything is decoded within the dynamics of the system, or is this just kind of, an artifact of the data? So that's kind of what they're getting at here. And their answer, of course, is that the model that they're showing here, the results that they're showing here, suggest this type of sort of pre-patterning and then amplification of the pre-patterning. And this is done by these bioelectric mechanisms. So this is a very busy paper. There's a lot of data in here, a lot going on. Looks like Dick had to leave, but yeah, this is a very big paper, and I think it, kind of gets into some of these issues, not only of bioelectricity and its role in morphogenesis, but also, kind of what it is to be sort of a self-organized entity. What are the steps that involve, getting these sort of self-organized structures in place? And so they answer that by, kind of laying out this model, doing some simulations, and giving us, I think, a lot of food for thought. So let me go to the chat here. We have some things here. I think Dick and Susan, yeah. So, okay. Dick said back to about 1900 and fish eggs. I think he's referring to one of the earlier things that I was talking about. Susan says, do they take into consideration the electric field in mitochondria? Yeah, they didn't mention mitochondria. I think this is more of a generalized electric field in the cell where you get these ion channels that are set up. You get this ion channel flux in the cell, and they provide this balance. And then you measure sort of the overall electrical activity. So whatever ion channels are expressed in the membranes, that allows for ion channel exchange, and then that gives you your
large electrical gradient surrounding the mitochondria.
Yeah, they don't get very much into intracellular stuff here. This is just largely intercellular stuff. So Dick says, sounds like gobbledygook, And then I know he's not a fan of the morphogenetic field stuff. So and then Susan says, put the electric field information into COMSOL and model it. Well, that's something. Yeah, I mean, like you could do that. I don't know what program they use for this. I'm not sure they're using like a standard program. I think they're just setting up these simulations. There is there's a lot more to the paper, first of all, and I don't want to give it like a short shrift in that sense, but they do set up these simulations. And it's not clear what they're doing, how they're doing this.
No, I'm multitasking here, Bradley, but which paper is this? Is this a Levin paper?
Yeah, this is Levin and Manicka. This is a new preprint that they have. Let me get the title for you.
Yeah, so previously they have their own software for electrical bioelectric cellular modeling.
Yeah.
Yeah, it's called Betsy.
Oh right, yeah, Betsy.
Yeah, it's really cool. I mean, and yeah, they developed their own tool to do that kind of calculation.
Python.
It is well, I think I wrapped it in Python. I can't remember now if the original is in C++, but I do think it's Python. But check Betsy.
Yeah, it's Betsy with 2 E's at the end. Yeah, it says. I remember we talked about this. I think it might have been in the Saturday meeting, but they do have this specialized software for this.
How do you spell that?
BETSEE.
There's some
BETSE.
So the paper title.
There's 2 of them. I forget which one was the earlier one, which one's the later one. Okay. But there's a second. Sorry, I'm in the car, so I would like totally look.
Oh, so the paper title is Field-mediated Bioelectric Basis of Morphogenetic Prepatterning, a Computational Study. And this is just a new preprint that came out. So
That sounds awesome. I mean, that like soon as I get kids dropped, I will be checking it out.
Yeah, it's a huge paper, so I just wanted to cover some of the stuff in it. And we talked a little
Oh,
bit
really?
about, yeah.
It's like when you say huge, like
Well, there's a lot of work in it, a lot of complex graphs you have to dig into. I mean, I
Yeah.
showed some, but I didn't talk about like their significance. So yeah, it's like something to follow up on
okay,
for sure.
okay, awesome.
Yeah. Now we talked about synergetics, which was interesting. I don't think they talked about it I don't think they actually were using like
Okay,
the
well,
Hay conversion,
yeah,
but there's definitely this aspect of stigmergy and things that we've talked about with respect to emergent systems where you have
sure,
emergence working, and so I think they lay out the
sure,
sort of the emergence aspect well too, and it
sure.
kind of
Sure.
gets at
Well,
some of those. Yeah,
you're mentioning Edelman and
Danoni and sporins. Yeah,
more. Okay, like they're not exactly Hawkins school, but very close. Kind of synergetics.
yeah. Oh yeah, they have kind of the stuff that they're doing with like integrated information and all of that.
Yeah. It's Yrsa's more like Yrsa was an actual student of Hawkins.
Okay.
Um, but like they're all coming at it from this kind of similar kind of biophysics
Yeah, it's
approach. Yeah.
it's the same math and everything. Yeah. Okay. So that's what we have for today. Now I know people are here; they might be interested in GSOC, so we haven't talked about that yet today. But we'll be continuing with projects. People have project ideas. You're free to present them in the meetings.
Just muted yourself.
Oh, okay. So yeah, if you want to present a project idea, you're free to present those in the meetings. You just tell me beforehand, we can reserve some time for you. Looks like we have a message here. I'm trying to get to the top. Aniketh, this is one of the people in the meeting here. Hey guys, I spent the last week going through the Graph Neural Networks code base and getting familiar with stuff like NCA and CMAES, since I was not really familiar with them. That's the stuff with neural cellular automata and the stuff that was forked last summer for the Growing Neural Networks project. So this is there's a lot of these things were forked from other places, so you have to get sort of some background in that to really work with it effectively. Still stuck a bit on how the training loop works, but I've reached out to Paki to help me with it. Yeah, Paki, of course, did the Growing Neural Networks, and Mehul did the hypergraphs. So if you're interested, whichever one you're interested in, Paki did one and Mehul did the other. But they're always willing to help out, at least to answer questions in terms of what they did last summer, their experience. So please reach out if you want to. If, they should be able to help you out. I wanted to try and work on this issue in GitHub. This is DevoGraph issue 27, which is coding oriented, and it seems to be stagnant as well. I hope I'll be able to close that by this week, and if it ends up getting assigned to me. Yeah, okay. So this is the issue here. This is DevoGraph issue 27, enhance and fix graph implementation for conformity and functionality. So this was opened on December 9, and I don't know who this opened this issue. I wasn't familiar with. I don't remember seeing this. It must have come up. I didn't pay too much attention to it at the time, but basically, um, the graph class in our repository, DevoGraph growing GNN graph.py, requires several enhancements and fixes to ensure better functionality, robustness, and API conformity. So it goes over the expected behaviors, and then the proposed solution and additional information. So yeah, issue 27 in DevoGraph, I think that's, or in in DevoGraph, I think that would be good to address if you can. Please, follow the sort of the way we do things, which is, you can issue a pull request, and then I'll take a look at the pull request. Just make sure that you document what changes you're making and how you're addressing the issue, because I'm not always familiar with a lot of the stuff going on in the repository. I'm not hands-on in that repository, or as hands-on as I'd like to be. So, yeah. So that sounds good, and that would actually be a good start. Maybe you could build. I don't know what you're interested in doing in that project, but the idea basically in these proposals is to propose something that you think would be useful to any aspect of this. So you can combine growing graph neural networks and hypergraphs, where you can focus on one or the other. You can focus on extending the results, or you can focus on some sort of maintenance. It just has to be long enough to be a GSOC project. And how long that is hard to say, but you do go through sort of the duration of GSOC, which is about 12 weeks, and you figure how much work would be good for a single week. And you just kind of make that calculation. And then that's how much work you would propose. So if it's just like a simple fix, solving a simple issue, if the issues are really simple, that's fine. That's probably not a GSOC project on its own. But if you extend that out and say, okay, let's build from that, and let's try to, maybe knock off a couple more issues that might be needed, or maybe updating things in the repositories, or you just have to think about the right amount of work for that. And usually I let the students kind of provide their own solutions or provide their own sort of guidance on that. I'm always free to look at project proposals to see if they're good, see if you're, that you've kind of gotten it in the right format, that you're asking the right questions, or that you're doing the right amount of work. So please don't hesitate to reach out. Okay. Yes. All right. Thank you. All right. So if there are no more questions or comments, I think that's it for today. Again, yeah, let's follow up on this Levin and Manicka paper if you want. And I know with Morgan, I wanted to remind him that I had brought up this business about organoids and how this is something that could be useful in organoid research.
I got the paper as soon as I get to get to a desk. Yeah.
I think Lilith had something to say.
Sir, actually, could you please share that modern artificial neural networks is evolutionary clear, that paper?
Yeah. Actually, I'll share it in the Slack, and I'll share it in the description of the YouTube video, which I usually do. But I can share the actual paper in Slack, because it's a neural computation paper, may or may not be accessible.
Yeah. Yes.
All right. Well, thank you for attending today, and talk to you next week.
Nice.
Bye.
Thank you.
Take care.
Yeah.